Hello class. So this is the second part of protein turnover and amino acid catabolism series. So the topic that we will discuss this time is the first step in amino acid degradation is the removal of nitrogen, and then the ammonium ions are converted into urea in most terrestrial vertebrates. So we're going to talk about the urea cycle that I had mentioned previously. So, as far as amino acids degradation, the first step is the removal of nitrogen. And so, this removal of nitrogen is done in the liver in mammals. And also, just note that muscles also readily degrade branched um, branch chain amino acids, and so there are examples that are listed here. And then also just as far as structure of what's going on, you have amino acid and you have an alpha amino group that is right here. And so it gets transferred to a alpha keto glutarate to form glutamate. And then from there, there is a glutamate dehydrogenase that releases the ammonia and reproduce the alpha ketoglutarate to form to be ready to be transferred again with some amino acids to form some more glutamate. And so this glutamate dehydrogenase is inhibited by GPT and stimulated by ADP. So in general, where you have this amino group transfer, it utilizes an enzyme called aminotransferase, which transfer the alpha group from the amino acid where it gets transferred on to a alpha keto acid. And so how is originally this alpha keto acid is formed? Well, this is due to this prosthetic group called PLM or pyridoxal phosphate group, which is described here. It actually has two different forms. So a tautomeric form where the hydroxyl group loses a hydrogen and not hydroxyl group. Yeah, hydroxyl group loses the hydrogen and is negatively charged. And the nitrogen group in the pyridine and so this whole structure right here is what they're referring to as pyridine. It gets protonated and have a positive charge. This overall structure is called phenolate. And so once it gets to this form, there is another enzyme that has a lysine amino acid residue that forms a shift-based linkage to um, to the um, to the PLM to the phenolate, excuse me. And then from there, you can have an amino acid that gets replaced. And so now that lysine that's from that enzyme is released, and another amino acid is covalently attached to the phenolate. Then from there, there's some restructuring that's happening. There's some deprotonation and protonation. And then finally, you have hydrolysis that happens that ends up releasing the alpha keto acid. So that is generally where gets released. and the other product is PMP. So once um, this is released, the alpha keto acid, it reacts with another amino acid to end up forming an alpha, ends up forming glutarate. And so with the glutarate, that is where we're at here. And so that's when the glutarate um, dehydrogenase happens, where you have these cofactors NAD plus and NAD P plus. You can use either or in your body, and it forms this intermediate ketamine. And then from there, the amino group is readily released to create ammonium, and this is toxic to our bodies, and so it is immediately uh, eliminated by having it being converted to urea, and then from there it's excreted from our body. Thus, once what pulls this reaction forward is this rapid release of ammonium, and it forms this alpha ketoglutarate that can now have another amino transfer to the alpha ketoglutarate to form the glutamate all over again. And so just in general, just looking at this cycle, you have an alpha amino acid and you have an alpha ketoglutarate that reacts with each other via the amino transferase, produces that glutamate and produces more alpha keto acids. And then from there, the glutamate goes through the enzyme glutamate hydrogenase. And remember, you have that NAD plus or NADP plus, either or, to end up producing NADH, which shows here on the product side, and then reforms that alpha ketoglutarate that can recycle again to make another glutamate. But with the other product for the NADH, you also have that free ammonium ion, and then that will eventually go to produce urea, which is then excreted out of the body. So, as far as the muscles and like breaking down the branch amino acids, so you see here we're talking about the muscles. You have branch chain amino acids. They can go to two different paths. One be broken down to carbon skeletons for cellular respiration, or it can transfer over its amino group to an alpha ketoglutarate to form that glutamate again. And then from there, how it can be used to actually be synthesized to make glucose is you have pyruvate reacts with the glutamate to form alanine, and alanine actually goes outside the muscles into the bloodstream, and then from there, that alanine gets broke interacts with the alpha glutarate once again and form glutamate and also pyruvate. And so that glutamate gets broken down once again with glutamate dehydrogenase to release that ammonium ion to eventually create urea and produces more gluto alpha ketoglutarate. But that pyruvate that ends up being formed can form the glucose And this all is happening within the liver, and then it goes back into the blood system, and then from there go back into the muscle and start the process all over again. And you form the pyruvate by glycolysis. So this urea cycle that I've been mentioning that is coming, this is it. So that free ammonium ion that gets released, it reacts um, with bicarbonate. And this bicarbonate, this comes from a hydration of carbon dioxide. And so what it does is they react together to end up making this carbamoyl phosphate, which reacts with the ornithine. And this is all happened in the mitochondria and produces these citrulline. And then from there, the citrulline goes outside of the mitochondria, and your actual urea is being created in the cytoplasm. And so what happens when the citrulline goes out into the cytoplasm, another amino acid that was created uh, by a amino group transfer creates this aspartate. And then from there you have arginino succinate, then produces the fumarate that's broken down, which is the skeletal to actually create more aspartate, but we'll get into that later. You'll have arginine as a product, and then with water, you have that release of the urea, and then produces the ornithine to start the process all over again, because the ornithine goes into the mitochondria and reacts with the carbamoyl phosphate group. But how do we make this carbamoyl phosphate group? So you have the bicarbonate. It goes through an ATP-driven type of reaction within this enzyme. This called carbamoyl phosphate synthase 1. And you have this phosphor group that is attached. Then you have that free ammonium ion that replaces it. And then you have this intermediate. And then you utilize some more ATP to transfer the phosphor group to this opposite side where you have the hydroxyl group, and now you have that final form of the carbamoyl phosphate group, phosphate compound. So now we have this carbonyl phosphate group. We need to talk about how does it react with the ornithine to create the citrulline. And so what happens is you have another enzyme called the ornithine transcarbamylase. So pretty much it's a condensation type of reaction. And then you have the carbamoyl group um, that ends up being attached to this the amino group of the ornithine to form this citrulline, and this citrulline is then is transported out of the mitochondria into the cytoplasm. And so that's what we see right here. And from there, we'll do the next step of the citrulline combining with aspartate. And so with the citrulline combined with aspartate, it goes through another enzyme reaction that is driven by the cleavage of ATP into AMP and having the pyrophosphate. And the enzyme is argininosuccinate synthetase. And then you have this argininosuccinate compound. Then from there, then you have this cleavage that is happening to end up forming the fumarate, and then you have your arginine. And so one of the other things that can happen as far as that fumarate creating actually glucose. What happens is we already talked about the carbon dioxide being hydrolyzed to be bicarbonate, reacting with the amino ammonium ion to form the carbamoyl phosphate. It goes reacting with the ornithine in the mitochondria, produces that citrulline again. That citrulline goes to the cytoplasm and reacts with the aspartate to produce that argininosuccinate. And then that argininosuccinate, you remember again, you have that cleavage where you have arginine and that fumarate. The fumarate is hydrated to malate, and then from there is oxidized to oxaloacetate, and then from there produce glucose. But when it's oxidized to the oxaloacetate, it's also producing 1-NADH, which can be used to generate 2.5 ATP molecules. And then from there, another option that the oxaloacetate can do is the amino acid can transfer over to the oxaloacetate to actually reform the aspartate, and you have this alpha keto acid, and then from there goes through that cycle again, and we'll continue on with the arginine, and where it eventually forms urea and that starting material, the ornithine, to start the process all over again. So we have this arginine. It goes through arginase enzyme activity with water, where this urea group ends up being cleaved off right here. It forms urea to be excreted through the body, and you have that starting ornithine that goes into the mitochondria and starts the process all over again. And that is the urea cycle. So this actual figure, you should start to become really familiar with it because it's one of the central source of how to understand how urea is released from the body, and when we do produce that ammonium ion, how is it being excreted out to the body? It goes through many steps, but these are the steps that are necessary in order for the whatever toxicness is in our body is being released. And that is part 2.